The next speaker is Vincenzo Cantaluppi, professor of nephrology, University of the Piemonte Orientale, UPO, and he will talk about the emergency concept of the tubular functional reserve. Okay, Vincenzo.
Thanks, Chairman, and thanks, Ravi, for the kind invitation for this 30th anniversary of the meeting. So this is the agenda of my presentation. We first look at the definition of kidney and dynamic stress test to look at what we can define as tubular stress test together with glomerular stress test and see the idea of the expanding concept of tubular functional reserve. We know from the nature what stress is and also stress test. This is what our friend cardiologists do every day. But when we talk about kidney stress test, things become complicated because always the kidneys are complicated. And so we have to distinguish between glomerular and tubular stress test. Probably glomerular stress test is the most used. It's based on the concept of renal functional reserve, GFR, that basically is the difference between the peak GFR under stress and the baseline GFR. So two different patients may have the same GFR, but different renal functional reserve. And we can investigate this functional reserve with an oral protein load. This is what we do in our kidney transplant unit in Novara in Italy, with this oral protein load with the, what suggested by the Claudio Ronco in the International Renal Research Institute in Vicenza. So after protein load, you can see that the renal functional reserve is somehow correlated with the increase of glomerular filtration rate in living kidney donors 1 year after donation. So higher is the renal functional reserve before donation, higher is the increase of glomerular filtration rate after 1 year. And the same concept always from Claudio Ronco group in Vicenza, in cardiac surgery patients, so lower renal functional reserve is associated with an increased incidence of AKI post-cardiac surgery. And in another group of patients subjected to cardiac surgery, you can see that patients who developed AKI after cardiac surgery show lower renal functional reserve. But the idea is, as suggested in this cartoon, for COVID-associated AKI, but for different episodes of AKI, may consume renal functional reserves, so facilitating the progression from AKI toward chronic kidney disease, as a sort of acceleration of aging. But the question here is, what about the presence of acute tubular injury? Most of AKI episodes are due to tubular injury and not to alteration of GFR. We know that tubular cells are very important. They do a lot of things, including they are immune active cells. They are able to antigen presentation. They are involved in the elimination of inflammatory mediators, activation of the complement cascade, release of ATP. So there are different things that tubular cells do. So we have tubular stress tests that we can perform in our patients, and some of these tests are based on the idea, on the concept of proximal tubular secretory clearance. So the idea that some of these molecules, including furosemide, for instance, may be absorbed by organic anion transporter present on tubular epithelial cells, in particular furosemide for OAT1. And the furosemide stress test is probably the most used tubular stress test. This is the paper from Milk Chavla some years ago, showing that furosemide stress test may identify AKI in looking at the urinary output 2 hours after the administration of furosemide. And these results have been confirmed. This is a meta-analysis showing that furosemide stress test, the accuracy of furosemide stress test in identify patients with AKI progression and the prediction for renal replacement therapy. A similar thing is also in the kidney transplant setting. We know that delayed graft function, DGF, defined as the need for dialysis in the first week after transplantation. And with the same test, the furosemide stress test, you can see that after 2 or 6 hours of stimulation, basing on the urinary output, we can identify patients who will develop or not DGF. And another possible molecule is indoxyl sulfate. Indoxyl sulfate is an uremic toxin, so in this case, binds with, it's absorbed through the OAT3. And what we know about indoxyl sulfate. Indoxyl sulfate comes from the gut, is a protein-bound uremic toxin that can reach the kidney and is excreted via urts. So the ligand is the aryl hydrocarbon receptor, AHR. This is poorly filtrated by dialysis membrane and progressively increased with the deterioration of kidney function. We know that in chronic hemodialysis patients, the levels of indoxyl sulfate are very high, but also in AKI, as you can see here, we have an increased concentration in the plasma of these protein-bound uremic toxins. And the idea is that indoxyl sulfate may act on the aryl hydrocarbon receptor located on tubular epithelial cells, inducing, as you can see, fibrosis and what we can define as tubular epithelial cell senescence. And as demonstrated by the Creek trial, if you see clearance of secretory solute, in particular indoxyl sulfate, is significantly decreased with the progression of chronic kidney disease. So the idea is now to have a sort of functional reserve of tubular cells, not only looking at the furosemide stress test, to look at other molecules or to what we can define as the maladaptive repair in the kidney. So some of the maladaptive repair mechanisms are correlated with alteration of tubular cell function, including the excretion of some well-known biomarkers of injury, such as KIM1 or NGAL. But I want to focus on the aging of the kidney, so how tubular epithelial cells maladaptive repair may act on kidney aging. These are the hallmarks of aging, not only referred to the kidney, but when we talk of kidney aging, I want to focus on these two, cellular senescence and mitochondrial dysfunction in tubular epithelial cells. Senescence is a very important and well-defined mechanism of damage. To stay young, kill zombies, because senescent cells are able to release inflammatory mediators. This is the so-called SASP phenotype that can induce damage within the kidney. And the idea is that during this maladaptive repair, we have an alteration of different molecules inside tubular cells, in particular these cytoprotective genes, such as NRF2, that is the most important antioxidant gene. So the activation of NRF2 is a promising tool to limit AKI to CKD transition. And we have different molecules here to explore this concept of tubular functional reserve. Probably in tubular cells, the most important is Cloto. Cloto is able to act on NRF2, so it's an activator on NRF2. And as you can see in the slide here, if you have a decreased expression in the kidney of Cloto, you have an increased staining, an increased histology for interstitial fibrosis and tubular atrophy. So decreased expression of Cloto promote what we can call tubular epithelial cell senescence. And this is already observed also in experimental model. This is an experimental model with ischemia reperfusion injury in swine, and also, once again, in transplant patients with delayed graft function. As you can see, Cloto is significantly decreased in its expression in patients with DGF. And the other hallmarks of aging is the mitochondrial quality control. We know that tubular cells, the balance between mitochondrial damage and mitochondrial biogenesis is very important to limit what we call the maladaptive repair of tubular epithelial cells. And in particular, this protein, that is PGC1 alpha, that is involved in mitochondrial biogenesis. And we know from experimental studies that PGC1 may promote recovery after acute kidney injury in mice with systemic inflammation in experimental septic models. And once again, in DGF in kidney transplant, as I showed you before for Clotho, also the lower expression of PGC1 alpha is associated with the development of delayed graft function and with an earlier loss of kidney graft function. PGC1 alpha is particularly important in mitochondrial biogenesis, as I told you before. And the idea is that during maladaptive repair, we have decreased expression of PGC1 alpha, but is also important to maintain this other molecule within the kidney, within tubular epithelial cells, that is NAD. NAD is critical role in the generation of ATP, as you can see here, and in particular in renal tubular cells that are highly metabolically active. And we can say that in experimental model of AKI, various insults may lead to decreased levels of NAD. And this is a very landmark study from Samir Parikh group showing this interesting ratio between quinolinate and tryptophan that is a biomarker of the concentration of NAD within the kidney. So the higher is the ratio, the lower is the NAD within tubular cells. And so the suggestion if transient NAD biosynthetic deficiency is a risk factor for AKI, NAD augmentation could be beneficial through the administration of nicotinamide or looking in particular at this de novo biosynthesis of NAD to block some specific enzymes, such as showed you in this study, the ACMSD inhibitor that is able to modulate the de novo synthesis, increasing the level of NAD into different experimental models of cisplatin-induced AKI or ischemia reperfusion injury. So what we can say is that the modulation of senescence and of the mitochondrial quality may be important to maintain what we can call tubular functional reserve. I go to the end of the presentation with a few data on extracellular vesicles that are different families of microparticles released by activated cells. This is not important, but the idea is that we have some biomarkers such as proteins and genetic materials that we can look at in the vesicles in the urine. And this is a very recent study in which experimental study, they see that the concentration of urinary extracellular vesicles correlated with the kidney volume. You can see how the excretion of extracellular vesicle decrease in animals subjected to unilateral nephrectomy or 5/6 nephrectomy. And this is also true for in humans with differences between male and females. But as you can see, the quantification of EV in the urine correlated with GFR or creatinine clearance. And last, we can also apply the omics technology to extracellular vesicles to search for protein or mRNA, macroRNA inside the vesicles. And we are studying these two molecules. One is CD133, that is the stem cell marker that can induce tubular regeneration. And so looking at the CD133 expression, we can identify patients after AKI that are more prone to progress towards CKD. And the last is the presence of Clotho, the anti-aging molecule. As I told you, you can see that we can also follow in extracellular vesicles, so low levels of Clotho in the vesicles, once again, are associated with a progression towards CKD. These are my take-home messages. Kidney stress tests are used for a dynamic and more complete evaluation of renal function. The combination of tubular and glomerular stress tests may potentially increase the sensibility to identify CKD progressors. And we have some new biomarkers, senescence biomarkers, mitochondrial health biomarkers, and urinary extracellular vesicle, as I've just shown you, that can contribute to this concept of estimating the tubular functional reserve. Thank you.